Thank you, Tejaswini. Now, as you know that we have to heat fix our smear. So, but you need to remember that you can't overheat fix your smear because as overheat fixing will lead to the distortion or the shrinkage or the morphology of the bacteria can change when you overheat fix it. So please don't do that. Prepare a good smear with a good heat fixing, and then we can move on to the staining techniques. And today I'll be explaining you these four staining techniques, which are simple, negative, gram staining, and acid fast staining. I'm sure that most of you have done any one or at least all of them in your laboratory. So now I'll be dealing with simple staining. In simple staining, as the name suggests, the staining is very simple because it just requires a single stain. And in this staining, you will understand your bacteria has a circular shape or a rod shape or a spiral shaped bacteria it is. You will understand that as you can see in the picture. And other than that, you can also understand if it's in chains, if it's a doublet, triplets, or like that. So you can see in the right side how the bacterial formations are there of different types of bacteria, so you can identify that. And for this staining, as my friend explained, basic stains, so basic stains have positively charged chromogen and they can easily penetrate into the cells, so they will stain your cells and they will make it colorful, and then you will understand the morphology and arrangement of your cells when you keep it under the microscope. So this is what you will see under the microscope when you do a simple staining. The left one, as you can see, it's diplobacillus. So you will be able to identify that if it's in chains or if it's doublet, triplets. Okay, so like you will be clearly seeing that in 100x, of course, like that. Okay, like one or two or three or four or five clusters are there or chains are there like that. And the right side, it's a spiral shaped bacteria. So the procedure for simple staining is very simple. You just need to prepare a smear and just heat fix it and apply crystal violet or methylene blue or whatever basic stain you have, and then keep it for 1 minute. Wait. You can just use your stopwatch, and then you just wash it under tap water, but make sure the tap water is not very, I mean, directly on the smear, as it can again wash off the smear. You just put drop wise of water in one end of the slide, and you can just wash it properly. Then you can air dry, or with the use of a blotting or bibulous paper, you can dry your slide. Then you can put it under microscope and you can see it. Okay, now we come to negative staining. So it's just the opposite of simple staining in simple terms. Basically, what it does is this stain will stain the background of the slide, but not your cells. Okay, and this is one stain. Please keep in mind, this is the like one of the stains in which we don't heat fix the bacteria. No heat fixing is required in this type of staining, and in this we use India ink or nigrosin. As these are acidic stains, like we will have to use acidic stain so that it doesn't penetrate the cells and the only the background is stained. So the acidic stain will only color the background, and you will be able to see the morphology of the cells as can be seen in the simple one as well. And also, other than that, you can see capsules. If, as we know, some bacterial species have capsules present outside the cell wall, so the capsule will be easily visible in negative staining. So negative staining is generally done for morphology and to understand if capsules are present in your bacteria. So one thing which you need to keep in mind is the precaution, like taking precautions, because as we know, we are not heat fixing this in this staining. You are dealing with the live microbes, so you have to make sure that you are wearing gloves and you are taking proper precautions before dealing with this staining, because you can get infections and everything. So please be safe. And this is the procedure for negative staining. This is slightly different from the other stainings we have learned. This in this you have to put one drop of India ink or nigrosin at one end of the slide, and then with a loop you can take a loop full of bacteria and you can spread it evenly with the loop. You can mix both the stain and the culture. Okay, and then what you will do, you will need two slides for this technique, and you have to use both the slides, like should be equally clean and grease free. So with the second one, you will put it in 45 degrees and you will prepare a thin smear, like you will just push the slide and prepare a thin smear. But here you have to make sure that the, like, because the India ink and Nigrosin is very dark in color, you have to make sure that the smear you prepare after pushing the second slide should be very thin, because like the amount you take in the first the first point is very critical, because if you take larger amount of Nigrosin or India ink, the smear will be very thick. But what will be the problem? The light won't be able to pass when you keep it under the microscope. So that one problem you can face in this method. So please make sure that the smear is thin, and light can pass when you keep it under the microscope. Okay, so when you keep it under the microscope, you will see something like this. This is a rod shaped bacteria, and it can be easily seen that it has chain formation. So you can easily distinguish between the shapes and the morphology when you keep it under the microscope, and check it in 100x, not 40 or 10x. So yeah, this I hope that the simple and the negative staining is clear to you. Please ask if you have any doubts. If you have any queries, please don't hesitate to ask us. If you like say yes, we have some, I mean, we don't have any problem, I will be moving to gram staining. Please respond. Okay, I think
Okay, we can go ahead.
yeah. Okay, so next is gram staining. This, as we all know, we have all done or at least read it somewhere or the other, but we don't have the proper concepts. Like sometimes we don't know why do we use crystal violet or what it does with the cell and everything. So I will explain you that today. And this method was developed by Dr. Hans Christian Gram. So in this, before moving to staining technique, first we have to understand what is gram positive bacteria and what is gram negative, and how are the different. How's the cell wall of both of this bacteria different? Okay, so as you can see in the gram positive bacteria, this the purple layer here is the peptidoglycan layer. So this is very thick, and it doesn't have any other layer outside. Above the peptidoglycan layer. So this is the final layer, and this is very thick in the case of gram positive organisms. But in the case of gram negative organisms, as you can see that this peptidoglycan layer is thinner when compared to gram positive. Plus, it has some other layers also, which are known as LPS or the lipopolysaccharides above the peptidoglycan layer. Okay, so what happens is, due to this difference, we can see that okay, the cell is taking this color and this cell is taking the other color, so which I will be explaining you. But the first and foremost, you have to understand the basics of cell wall of both the bacteria. So what is peptidoglycan here? Peptidoglycan is like in both the cases, peptidoglycan is made up of NAG and NAM complexes, which are N-acetyl glucosamine and N-acetyl murimic acid. So these are cross-linked using a transpeptidase enzyme. Okay, in this purple layer, these are cross-linked with one another using transpeptidase enzyme, and this gives rigidity to the cell wall. So for staining purpose, we are okay if we know this, but like only this thing like peptidoglycanol. If you want detailed understanding of the cell wall, so we can we can do a upcoming webinar on that if you wish. Please let us know in that case. So now we will be dealing with only the peptidoglycan layer and the other above the layers which are present above the peptidoglycan in the gram negative bacteria. Okay, so now what, these are the four stains or the reagents we use, not the stains, sorry, we use only two stains, but there are four different steps in gram staining. So the first one is the application of crystal violet. As we all know that the crystal violet is a basic stain, so it will be able to penetrate to all the cells, irrespective that it is gram positive or negative. It will enter all the cells and it will stain all the cells purple. Okay, so the second step here is the application of grams iodine. What does the grams iodine do in this case? It acts as a mordant. The word mordant means it enhances the ability or the cell's affinity for a stain, which means it will enhance, in this case for the crystal violet, it will enhance the cell, it will make the cell more permeable so that crystal violet can be uptaken. And then what it will do, the crystal violet and the iodine together will make a CVI complex, which is known as the crystal violet iodine complex, and that is insoluble in water. Okay. In gram positive organisms, what it will do is, like, it will form the CVI complex in both the gram positive and the gram negative, but in gram positive it will be retained, and like it will be not easily washed away when we do the next step, that is the decolorization step. But in the case of gram negative organism, it will be washed away easily. So in this case, if you, in this step, if you keep your slide under the microscope, you will see that all the cells are purple black in color. Even if it's gram negative, it's gram positive, everything will be appearing purple black. So the next step is the decolorization step, in which we use 95% ethyl alcohol. So guys, please make sure that you don't mess up in this step, because this is the most crucial step of gram staining. Because this, if you decolorize for a longer period of time, all the cells will leave their CVI complex and will appear colorless, or either after safranin it will appear pink. So you have to make sure that you do it properly. Okay, so what does the ethyl alcohol do? It acts as a protein dehydrating agent and as a lipid solvent. So I will explain you one by one. First, I will tell you what happens in the case of gram negative organisms. As I told you, that gram negative organisms have this lipid layers outside the peptidoglycan layers. So in this, after you add ethyl alcohol, these layers will be dissolved. Okay, these layers will be dissolved, and the cell's porosity will increase, and the CVI complex will be taken out of the cells. Okay, the ethyl alcohol will take out the CVI complex outside the cells in the case of gram negative cells. But because the gram positive cells has uh, thicker layer of peptidoglycan layer, so the ethyl alcohol will make sure that the pores are, made smaller, so that the CVI complex are not washed off when we are doing this step. So it is mandatory for you to make sure that you do the decolorization step very properly and very cautiously, because this is the step where the cells can lose their colors. So this is one step, and the last one is the counter stain, in which the safranin is used. Here, as the gram negative cells now don't have any colors, so they have the place to, okay, take safranin. But in the case of gram positive cells, they don't have any place to take up safranin, because they already have that CVI complex in them. So there's no place for safranin to enter. But in gram negative cells, the safranin is taken up. As you can see in this cells, they appear pink in color. I'm really sorry. They appear pink in color, and the gram positive cells appear purple in color. Okay, this is the actual image under the microscope. These are the gram positive cells. Okay, these are purple in color, and this is gram negative. They appear pink in color. So this is the overall gram stain procedure, which I already explained it to you. So every step is for 1 minute. You can keep like crystal violet for 1 minute, and then you can wash off. Then again, gram iodine for 1 minute. Then again, wash it off under the tap water, and then you have to add 95% alcohol. Okay, you can add it under running water as well, and you can see when the last drop falls off from the slide, it must be colorless. So in that scenario, you understand that, okay, my decolorization step is over, and then you move on to the counter staining procedure, which is saffron. And then again, for you have to keep it for 1 minute, and then you can air dry, and you can put it under microscope and observe. I hope the gram staining is clear now. So these are the precautions you have to take while doing gram staining. You have to make sure that you do the decolorization step properly, as I told you. And you also have to make sure that the culture you use for gram staining is fresh and not very old. Because what happens is, when you use old cultures, especially gram positive organisms tend to change their cellular morphology, and they will appear gram variable. Okay, they will appear gram variable when you stain them. So you will be perplexed that this was gram positive, and now like I can see both gram positive, gram negative, or something like that. So you will be confused. So in order to avoid that confusion, please make sure that you use fresh cultures while doing gram staining. I hope that the gram staining procedure and the protocol, everything is clear, and how the stains and the decolorization step or the application of iodine works when in order to gram stain. If you don't have any doubts, we can move to the acid fast staining next. Please, let us know that if you have any doubt. Okay, I think it's clear. Okay, this is the last staining I'll be discussing with you today. So, does anyone of you know that what is the other name for acid fast staining? Can someone tell me? Please don't be scared. Like, if you even if you answer wrong, you can answer wrong. No problem in that. Okay, I don't think. Okay, so the other name for acid fast staining is Zeal-Nielsen staining. Zeal-Nielsen staining, guys. Okay, please remember that. And like what happens is, now, as you know, that gram positive, gram negative, two types of bacteria. I have explained it to you, like the cellular morphology, the cell wall. Now we have another type of organism, which is the Mycobacterium species. So what happens in this organisms? There is a mycolic acid layer, wait, this is the mycolic acid layer. Here's the gram positive, gram negative, and the acid fast organisms. As you can see, there's a mycolic acid layer outside the cell wall. So this mycolic acid makes it difficult for the stain to enter, and the stains are not, I mean, the cell is not permeable to take up the stain. Okay, so we have to use a different method, which is the acid fast staining, to stain this kind of bacteria. They actually do resist the decolorization by acid alcohol solutions as well. So, I will just explain you the procedure first. So, what you will do is, first you will prepare a smear and heat fix it, and then you will flood. The first step of acid fast staining is flooding the slide with carbolfuoxin. Then you will wait for 1 minute, and then you will put heat, I mean, you have to heat it. Okay, for 5 minutes you have to heat it, and then what will happen is, the carbon will be easily permeable, I mean, the cell will be permeable, and it will take up the carbon. So here heat is the mordant, because as I told you, heat, I mean, mordant enhances the cell's affinity towards the stain. So here the primary stain is carbon, and mordant is the heat, because heat is helping the carbon to enter into the cell. So the next step here is the application of acid alcohol. Just like in gram staining, we have to decolorize in this acid fast staining as well. So here the the acid fast bacteria will not leave the carbon, but the non-acid fast bacteria will leave the stain. And then the last step is the counter stain method, in which methylene blue is generally used. So if you apply methylene blue, the non-acid fast bacteria will turn blue in color, because they will take up the methylene blue, but not the primary, I mean, the acid fast bacteria, which have the primary stain, which is the carbolfuoxin. So there's no place for methylene blue to enter. So you will just see non-acid fast as blue and acid fast as pink. As you can see in this picture, the pink ones are the acid fast bacteria, and the blue ones you can see here are the non-acid fast bacteria. Okay, so if you have any doubts, so you can ask us. This marks the end to our 2-day session on Introduction to Microbiological Techniques. If you have any doubts pertaining to yesterday's or today's session, you are free to ask us. Please interact with us. Uh,
Guys, if you have any sort of doubt regarding the webinar, please go ahead and ask us the questions.
and if you want me to sum up the entire, like, staining techniques and stains, we can also do that. So please let us know, like,
I'm having a question actually.
yeah,
So basically, in gram staining also we are doing like we'll able to see the morphology and all, and in the simple staining also we'll be able to see that. So what's the need of doing less simple staining instead of like we can do directly the gram staining instead of doing simple staining or negative staining? Like
yeah.
in both three of them we can see the morphology, right? So
Yeah, true. So basically, what happens in gram staining is you are able to distinguish between gram positive and gram negative organisms. This is generally done to identify your bacteria. Like, if you want to isolate some bacteria, you have to first identify that, like, for example, if you want to isolate a gram positive organism, okay, so you first have to check the gram nature of the bacteria, which for which you have to do gram staining. But if you just want to check that what morphology your cells or your bacteria has, you can just go for simple staining, because that's easier and that's take a lesser amount of time when compared to gram staining. So that is the difference between these two.
so basically, instead of doing the negative staining directly, we can directly do the gram staining and understand the three of them. It's like
Yeah,
whether it is gram positive or like even the structure also we can understand from right that
right.
right.
From negative staining, you can also understand if the bacteria has capsules, which is difficult in the case of gram staining or simple staining.
Okay, thank you.
Okay, any more doubts?
I have a doubt actually. By acid fast staining, are there any different methods rather
Different.
than carbon fast staining?
um Carbon fixation, I think, like many of the basic stains won't be able to enter in mycobacterium species. So carbon fixation is the most used stain in this case, because it, like, the when you heat it, the carbon fixation can easily enter. But if you use crystal violet or other basic stains, they won't be it will be hard for them to enter into the bacteria. So carbon fixation is the most used stain for acid fast.
Okay, thank you.
Yeah. Any more doubts? Okay. Do you want us to sum up the entire session?
Just react yes or no.
Yes. Okay. So Tejaswini, you do your part, I will do mine. I'll have some water by that time.
Okay. So in today's session, what we went through was one is bacterial staining, why we use a bacterial stain, and what does a stain contain, which is a benzene ring, a chromophore, and an oxochrome. Okay. Next is we go ahead with types of stain, which is acidic stain and basic stain. Saurav, can you change this slide?
Sorry, sorry, sorry. I'm so sorry.
It's okay. So we go ahead with types of stain, which is acidic and basic stain. As we said, acidic stains are anionic in nature, so they have a negative chromogen, negative charge on the chromogen. So they go ahead with having greater affinity to positive constituents of the cell. Whereas in our basic stain, these are cationic in nature. So the chromogen exhibits positive charge. And as chromogen is positively charged, they have greater affinity to the negative constituents. Okay. We have listed out the examples so that you can go ahead and see. There are like a lot of acidic stains and basic stains which we use in our laboratories. Okay. Next, we go ahead. We discussed about staining techniques. Okay. One is simple staining. Another one is differential staining. Okay. So, simple staining, we used one stain, one single stain, and in differential staining, we use two contrasting stains. Okay. So, simple staining, we usually, as my friend discussed, we use it for only morphological studies or arrangement of the cells. And differential staining, we use it for differentiating between the cells, which is gram positive, gram negative, or acid fast and non-acid fast. Or we use differential staining to visualize the individual structures of a cell. Okay. Next is smear preparation. What is a smear? How a smear is prepared? Okay, a smear. How what all you have to remember while preparing a smear, and why we need heat fixing while preparing a smear. I'll tell the reasons again. Okay, so one is because you need to heat fixing helps in denaturing the protein and easy absorption of the stain. Second one is it kills the bacteria, so you're safe from the harm of the microorganisms, and it is going to preserve the morphology of the cells. And the third reason is attachment to the slide. Okay, when you heat fix these smears onto a slide, there are lesser chances that the smear gets washed off. The bacterial cells get washed off. So these are the three reasons we need to remember while you are preparing a smear. Okay, then we go ahead with how we are going to prepare a bacterial smear, which is by two methods. One is broth medium, we can extract our sample, and from solid medium, we can extract a sample. Okay, broth medium, two loops of our sample, make a smear, air dry, and heat fix. In solid medium, you have to add one more step, which is addition of 1 to 2 loops of a liquid media, which is usually water, distilled water, and then we add small amount of our bacterial culture extracted from our petri plates or media, where the culture that we have obtained, and then prepare a proper smear, and then we go ahead with air drying it, heat fixing. Okay. I have one question because you guys have no doubts. I wanted to know if you guys know this answer. When we are doing broth medium, we don't go ahead with using this water, distilled water. But when we are going ahead with the culture and from a solid medium, we take 1 to 2 loop full of distilled water. Can you explain why we do this? Just guess, okay, why we are using water when we are going to extract from a solid medium.
So that the smear is evenly formed on the slide. I don't know, it may be wrong also.
It's partially correct. When you are making a smear with the help of a broth medium, there is there's already liquid. Okay, the solution is already a solution. Okay, so you can easily make a smear. But when you are taking a culture, it is almost just bacterial cells. So when you make a smear, it is not going to evenly spread. So you use water as a media for even spreading of the bacteria on the slide. Okay, next is
So the
staining
next
techniques.
staining techniques. So first I discussed about simple staining, which is done to understand the morphology and the arrangement of the bacteria. When it's not important to you to know that if it's gram positive, negative, whatever it may be, like then you can go for simple staining. It's very easy. You just need one stain. Any basic stain will do. Okay, you will just stain for a minute and then you will wash it under tap water, but make sure you don't wash it directly under the tap water. You don't put your slide, you just tilt it, put it in 45 degrees and then wash it and then dry it, and you can observe something like this, which is you can observe the arrangement of the cells. Then in negative staining, we don't heat fix the bacteria first thing. Second thing is we use two slides for this technique, and we can use any acidic stains. Okay, we can use any acidic stains for this so that it stains the background and not the cells, because the acidic stains are able to enter into the cells. Okay, and the natural size and shape can be seen by this method, and capsules can also be seen, but make sure you handle the slides very carefully. Okay, like I have one question for you guys. We can see the morphology in this one also, but in the simple staining also. So which one would you prefer? Like if I tell you that, okay, you just go and check the morphology of the bacteria, which one will you prefer? The negative staining or the simple staining?
So for seeing the morphology only, I would prefer the negative staining because, as you have already told, like in negative staining, we don't do the heat fixation steps. So basically, as she has already said that in heat fixation, the like morphology might get like changed during that step. So basically,
Yeah, that's absolutely correct. That's absolutely correct. We will have to go for negative staining because the morphology here won't change, because overheat fixing or heat fixing certain at certain point it will change the morphology. It will cause shrinkage. It can distort the cells. So if you want to see your cells as it is, you have to go for negative staining. That's an absolutely right answer. And then we move forward to gram staining, and I explained you the difference between the gram positive and the gram negative organisms and the cell wall of these both. And it has a gram positive has a thicker peptidoglycan layer when compared to gram negative, and there are certain other layers as well when it comes to gram negative bacteria. And also, if you want to know deep, like in detail, the cell wall morphology of both the bacteria and everything, we can do that. We can make it for you. Okay, the peptidoglycan has NAG and NAM complexes. Okay, and the gram stain has four different steps. The first one is crystal violet. Second one is iodine, which helps the cells to take up the crystal violet, and it increases the cell's affinity. And it also forms a CVI complex, which I already explained, the crystal violet iodine complex. And in the case of gram positive bacteria, the thicker peptidoglycan layer doesn't make it easy for the CVI complex to leave the cell when we do the decolorization step. And this is, as you now know, that this is the most crucial step of gram staining. And it also has certain functions which now you know, that it has a protein dehydrating agent when it comes to gram positive organisms, and as a lipid solvent it functions when it comes to gram negative organisms. Okay, so lipid solvent, what it means, it just dissolves the outer layers of the gram negative organisms, and like the CVI complex will come out. Because of that, the porosity, I mean, the porosity will increase, and because of that, the CVI complex will come out of the gram negative cells. And in the gram positive, it acts as a protein dehydrating agent, which means the pores will be made even more smaller, and the CVI complex will stay inside the peptidoglycan layer. So now you have to do the last step, which is the application of safranin, which is also known as the counter stain. Like counter stain is the second stain you are using. Okay, primary stain is the first, and counter stain, like in this case, it is safranin. It can be something else. Like for example, in acid fast, it is methylene blue. So that is also a counter stain. The second stain you are using is the counter stain. So here, the only the gram negative cells will take up the color of safranin, as there's no place in gram positive cells to enter, like for the stain to enter. So so the cells will appear something like this under 100x when you keep it. The negative staining of E. coli and the positive staining of streptococci. Next, you have to keep these things in mind, that you don't overly decolorize the cells, and you have to use fresh cultures, or your cells will appear gram variable. The last one was the acid fast staining, in which the mycobacterium species are generally stained because they have mycolic acid in their cell wall, and they don't allow all the basic stains to enter. There are only a few which can enter, one of which is carbylfluoxin, and you have to apply heat to enhance. And here, heat, that's why acts as a mordant, because it enhances the uptake of carbylfluoxin, which is the primary stain. And then you have to decolorize using acid alcohol. Acid alcohol here can be 3% acid alcohol. Okay, and then you have to go for the counter stain, which is methylene blue. And the acid fast cells now will look pink or purple, and the non acid fast will appear blue in color. So these are some pictures in which you can see both the acid fast and the non acid fast bacteria. Thank you so much, guys. If you have any doubts, again, you can ask us. From yesterday also, if you have any doubts, you can ask us from the media and the isolation of pure culture, from that. I hope you enjoyed the session today and yesterday, and you have learned, or at least revisited, those concepts you learned very early in your career, that is BSc, MSc. So that's a revisit of those topics. So please tell us that if you have liked our session, because your feedback is really important for us. And like, which you can also tell us, like which topics are you struggling with. We can plan an upcoming webinar on that. So please tell us, like if you have some preferences for the topics, like from microbiology, molecular biology, anything.